Now, the complete principle, like the pH meter will be made up of electrodes, yeah, we call it probes, which itself is made up of 2 electrodes. This probe passes electric signal to the meter, which displays the reading of the pH unit. So, the glass probe have 2 electrons because one is a glass sensor electrode and another is a reference electrode. And in many pH meter, both the electrode are placed separately, and in many cases they would in many cases sensor electrode and the reference electron, they both are placed together, and if they are placed together, then it is considered as the combination electron. Now, both the electrode are hollow bulb containing potassium chloride solution with the silver chloride wire suspended into it, and the glass censoring electron has a bulb which is made up of very special glass coated with silica or metal salt, and the glass censoring electron measures the pH of the concentration of H ion surrounding the tip of the wall glass bulb. So, the reference electron has a bulb made up of non-conductive glasses or the plastic material. So, it will be non-conductory, it is not going to conduct any electrode or metal ion through it, but the glass electrode it going to conduct, and glass electrode is responsible for the concentration of hydrogen ion. When one metal is brought in contact with another, a voltage difference occurs due to their difference in the electron mobility. Electron mobility means the meaning of electron mobility is the movement of electron from one electrode to another electrode, because they are going to start. So, every metal ion the electron presence in every metal ion are not having the same electron mobility, they have different mobility. Similarly, in the case with two liquids, a pH meter measures essentially the electrochemical potential between a known liquid inside the glass electrode and the unknown liquid outside. One KCl liquid is present inside the electrode and one unknown liquid is outside the electrode, and because of this the thin glass bulb allow mainly the small hydrogen ion to interact with the glass. So, the glass electrode measures the electrochemical potential of the hydrogen ion and the potential of the hydrogen. So, to complete the electric circuit, a reference electrode is needed. A pH meter is an instrument used to measure the pH of solution. It is a summary of the principle. It consists of a digital voltmeter which connect to a glass electrode. A glass electrode and a reference electrode are used in conjunction of each other to find the pH of the solution, and a reference electron that is built into the glass electrode is called a combination electron. These are the most common type of glass electrode we use. Basically, now what are the type of pH meter? We have two type of pH meter, that is manual pH meter and digital pH meter. This is manual pH meter, the pH meter which we are going to see. Now, the construction of pH meter, basically the combination of pH meter is of two parts, one is power pack and another one is electrodes. Now, what the things come in the power pack is the instrument. So, like we have to on the instrument, then it indicates the meter, there is a temperature compensation node from where we can adjust the temperature according to the room temperature of the room or on the basis of the temperature of the solution. And there is a calibration knob which is used for the calibration of the instrument and all the plugging wires. And what are the things which is come under the electrodes? So, one is glass electrode, one is calorimeter electrode. If your pH meter is of that type which contain two separate electrodes, then we will need two electrons. And nowadays in a digital electrodes they use a combination of electrodes. So, both the electrodes are present in a single electrode. Traditionally, a thing was used, a natural pH indicator which was known as litmus dye. What is litmus dye? A litmus dye is a litmus paper. It is a small strip of paper that contains dye in it. When it is touch a basic or a dye, then the litmus paper change color depending on either it is basic or it is an acid. If a litmus paper is exposed to an acid, it either changes its color to pink to red, and if the strip of litmus paper touch a base, then the paper changes to shade of blue. This is the pH scale which is natural scale of the things. Like this blue strip shows the alkalinity solution, in the middle it is a neutral which shows the water sample, and above is the acidic solutions. Above is the acidic solution. Now, litmus paper with a drop of base here, when it is having base drop, then it is going to turn into shade of blue, and when blue litmus paper with a drop of acid, then it is going to turn into red. Like HCl is highly acidic, it has 0 pH, lemon it has 2, so it is also acidic, apple 4, banana 5, water 7, so it is a neutral. Then we will come to the basic side, so baking soda, ammonia, these detergents, these are on the basic sides. So, HCl is highly most acidic, then vinegar, tomato salt, milk, these are the samples. Now, type of probes which we use, like we use glass probe, reference probe and combination gel probe. First one is glass probe, second one is reference probe. We will see the detailed diagram of these probes. This is glass probe which is contain of a glass membrane. There is an inner buffer of KCl and elements are present which are shaded inside it. This is a glass electrode. This is only responsible for the hydrogen ion activity. A reference electrode, it is made up of porous ceramic junction which is non-conducting material. Inside it, it has the silver electrodes, or if we are using mercurous chloride, then it is that one, and the reference element, and it is having a refilling opening. Whenever this liquid dries, then we can refill it. So, what is the popular reference system we use? We can use mercury calomel system or silver chloride system. And what is a reference electrode do? It is inert in nature, high ion concentration, and it contacts with the measuring solution. It is used for the completion of electric circuit. And now, latest which we are using is a combination electrode. A combination electrode is made up of two electrodes. One is glass electrode and reference electrode, both are combined together in one electrode system which is known as combination electrode system. The working of pH meter, what is the working of how it is going to work? The potential of the glass electrode is measured against that of the reference electrode by using the equation which is known as E is equal to E0 plus 2.303 RT A log A H positive, where E0 is the standard potential and 2.303 RT NF is the nest potential, that is slope factor, which change in the potential per pH unit we can measure, and it depends upon the absolute temperature. This is a circuit in which a glass electrode and reference electrode are put. When they come in contact, then they behave like a battery, and they start generating electric potential. This is the working. Basically what happened in this is that whenever this electrolyte come in the contact with the sample, they start initiating the hydrogen ion activity over there, and all the hydrogen ion come around, get surrounded around the electrodes, and there starts the exchange of the ions with the H positive ion, and due to difference in the electron mobility of the electrolytes present, they generate the potential, and this potential displays on the potentiometer. And there are the layers, these electrolytes are present between the inner gel layer and the outer gel layer. So, there is H positive ion is only the ion which can cross this outer gel layer.